Hello, good afternoon, everybody. My name is Bruno and I will be moderating this lecture today with my friend André. This broadcast is being carried out in a partnership with the seminar class coordinated by Professor Frederico Falcão Salles from our entomology program. Today, the theme is a bacterium in the fight against arbovirus. This is a topic of wide relevance and that has been gaining space more and more in entomological studies and control of disease vectors. And you are have the chance to talk to an expert on the subject. So make sure your question is in the live chat on YouTube because it is going to be answered after the presentation. As usual, today's broadcast will be divided in two moments. In the first moment, the question will be freely addressing the subject in the presentation. And in the second moment, we will dedicate the space to the public, and the guest will answer the public's questions.
Welcome, my name is André. Bruno and I will be mediating this broadcast. We have the honor to receive a very special guest today, Luciano Andrade Moreira, the coordinator of the World Mosquito Program Brazil and researcher in public health. Dr. Luciano is an agronomist by the Universidade Federal de Viçosa, master in phytotechniques with emphasis on biological control of insects by the Universidade Federal de Viçosa, and doctor in plant genetics and breeding by the same university and the Center of Plant Breeding and Reproduction Research, the Netherlands. He made a postdoctoral at Case Western Reserve University and another postdoctoral at the University of Queensland, Australia. Today, he is the coordinator of the World Mosquito Program, Brazil, as I said, and he's a researcher at Fiocruz in the field of molecular biology of arboviruses transmitting mosquitoes. Welcome, Luciano.
Hello, everyone. Thank you very much for the invitation to be here today. Thank you, André and Bruno and the sector folks for all the organization here. And it's an honor to be back to Viçosa, where I studied long time ago. I was a student there. I did my master's degree in entomology, linked to entomology as well. And and then I had the opportunity to visit other labs in the world and also be now a scientist at Fiocruz. It's an honor to be here today, and I'm gonna present the bacterium in the fight against arboviruses. And for me, this program is really an example of translational research. So I put these two pictures here for you to see. The first one is the lab where I joined in 2008 for sabbatical or a postdoctoral training. And there we discovered what I'm gonna show you next. And the right side photo show you what we've been doing in Brazil with schools. So trying to bring the program into schools and not only talk about Mobakia and mosquitoes, but try to put all the context of these scientific findings in their daily lives. So really translational research that I'm gonna show you now. The World Mosquito Program is currently in 11 countries. It started in Australia and went to other countries like Vietnam, Indonesia. I was able to spend two and a half years in Australia where we made some discoveries around this program. And then we brought the program to Brazil in around 2011. And later on, Colombia and Mexico also joined the program too. So 11 countries in this program. So what's the World Mosquito Program? It's a natural, auto-sustainable, and a not-for-profit initiative. We are not a company. In Brazil, it's conducted by Fiocruz and involves the releases of Aedes aegypti, which is the vector of dengue, Zika, chikungunya, containing Mobakia, which I'm going to explain to you now. The objective is to control dengue, Zika, and chikungunya, the main diseases that this mosquito transmits. Now I present to you Mobakia, which is an intracellular microorganism, so it's a bacterium. It's present in around 60% of all insect species. So all of you study insects, if you go outside, each 10 insects that you collect, I'm sure in at least 6 you'll find Mobakia being naturally present. So the way it works, Mobakia, it's inside the cells, and it's mainly positive in ovaries of the females. Females transmit the Mobakia through their eggs to all their offspring. So Mobakia manipulates their hosts to make sure that they will be transmitted to the next generations. So it's a great, makes a great manipulation of the reproductive. But other things were found that Mobakia also do. I'm going to show you next. Although present in many insect species and also mosquito species like Culex mosquitoes, and also Aedes albopictus, it was never found naturally in Aedes aegypti. So this video shows you how it was done in Australia. Mobakia was purified from Drosophila melanogaster, the fruit fly, and then introduced into Aedes aegypti eggs. And in this process, there was no genetic manipulation. It was a natural introduction of bacterium and into this mosquito species. There was no genetic manipulation either in the bacterium itself and also in the mosquito. So there were some findings after this, and I was very lucky to participate in the discovery that when Mobakia is present in Aedes aegypti, it blocks the replication of viruses that they normally transmit, like dengue, Zika, chikungunya, and then this mosquito will not become a vector. So here are some examples of publications in terms of dengue, chikungunya, and Zika, also Mayaro, and also yellow fever virus as well. So our objective again is to release Mobakia mosquitoes. So in this scheme here, this diagram, you can see wild Aedes aegypti population. So these mosquitoes in nature, they don't have Mobakia, as I mentioned before. So what we do, we release for around 16 weeks or 4 months, every week, a bunch of mosquitoes with Mobakia. So what happens? They will start crossing with the mosquitoes that are there. So we release both males and females mosquitoes. So, and then these mosquitoes will pass through, remember, Mobakia is passed through the females to the offspring. And the idea is then that Mobakia mosquitoes will be predominantly present in that environment, and that we expect to in the end to be mostly Mobakia mosquitoes being present, and then we can protect the population, human population, towards dengue, Zika, chikungunya. So that's the whole rationale regarding the program. We can do releases through adult mosquitoes like this, with just walking the streets with a cup full of mosquitoes, or through vehicles, I'm going to show you later on too, or eggs. We can install some buckets or these noodle boxes, tiny box containers that contain eggs, and the mosquitoes will fly out after 10, 15 days. So that's the 2 methods we can use. So what are the main steps? First, we have a period of communications and community engagement. So this is very, very important part of the program, because we want the community to be part of the program. We want to explain the program to really to take to solve all these questions that they have, and also to tell them how everything works. So the community really is engaged upon this the program, and they will participate. So just some examples of printed materials, some posters showing what we do, or what we can also touch around mosquito breeding sites in their homes and neighborhoods. We have some pamphlets discussing, explaining around the program, and then we distribute in some schools and also fairs that we organize with the community. Of course, with the pandemic, we are not able to do this anymore, but we've done that in the past. We have some facts for health agents. So this is our partners that we use in the municipalities that we work. So they approach the community members and they explain the program for us. So just to really to have more expansion and then more coverage of the program in each city. We work lots of digitals like WhatsApp and social media. Really, mostly now after the pandemic, we are using a lot for this for engagement, and we have videos for not only talking about the program, but also talking about the environment, to reduce mosquito population in their backyards, and some other examples. We work basically in three sectors. First, with the health sector. So here are some photos showing you the approach we get with capacitation of and training of health agents, both the ones that just do vector control, and also the ones that go, and the community agents that they get into people's homes and talk to them and talk about vaccines and so on. So we engage them, they are part of the process, so they help us in the process of engaging the population. The second is the community leaders. We identify in each neighborhood that's very important to help us to really to gauge how to calibrate how we are gonna work in that particular community. They are very important and very good partners for all our activities. And the third sector is our schools. Of course, now not every city schools are open, but we've done lots of activities for many different children, different ages, talking about the program, but also bringing the scientific question inside schools and doing lots of activities in schools. And the kids love, the kids bring this idea and the information home and talk to their parents, which really spread the word around the program. We have in each city, sometimes more than one, but a community reference group. So these are people that are from different areas in that particular city. So they gather together with our team members. So of course they are engaged and then they participate. So they are a way of really getting close to the community. So we bring information, then we take information from them to really to work in that particular territory. It's very important, the role of the community reference group for the success of our program. Now I'm going to talk about production, releases, and monitoring. So in these photos here, you can see some of our production in real. So blood feeding, production of eggs, and at the moment we produce around, let's say, 4 million mosquitoes per week, and around 4 million eggs per week as well. We want to go much bigger later on to have really a huge biofactory. That's on our plan now. So this is the release of Aedes aegypti with Mobakia that we do in most cities. So this is an example of in Rio de Janeiro. Really a vehicle driving around every 50 meters. They stop, they open a tube and release mosquitoes so they can fly away and mate and cross with the local mosquito population. In areas that we cannot get in because it's safety reasons, some areas like favelas and communities in Rio and Niterói that we worked, and also areas that we have very narrow streets, for instance, we work in partnership with health agents, so they can walk really with a backpack and have these tubes there that they can open in each release point for mosquitoes to fly out of these tubes. So these are the traps that we use for mosquito monitoring. So these are the BG traps. They collect adults. So it's linked to a power outlet, and there's a fan, and then mosquitoes are collected in a small bag. Every week we go to these places, people's homes, and also commercial establishments, and then collect these mosquitoes to bring to the lab and detect Mobakia. So we want to know if Mobakia mosquitoes are getting more and more prevalent in that specific area that we are doing the implementation. So just for you to have some numbers, in the peak activities in Rio and Niterói that I'm going to show you next, we used to have 1,300 of these BG traps installed in people's houses and commercial establishments. So it was a great logistic around this activity. And so bring it to the lab, we do molecular analysis, we do quantitative PCR to detect Mobakia in these mosquitoes, and then make sure that Mobakia is establishing accordingly what we expect in that particular area. So I'm going to show you some results, early results from Rio, the pilot studies. And so the two areas that we work in are called Tubiacanga and Ponto Final. These areas are quite small. There are around only 4,000 people that lived in each area of these ones. We did release this back ago in 2015. We stopped in January 2016. So now in January this year, 2021, it's been 5 years that we stopped releases in these areas. Since then, we are doing monitoring. We're still doing monitoring in these areas just to follow Mobakia integration there. So that's the beauty of, I'm going to show you this graph here. So just before, I'm going to show how is the area. It was kind of divided geographically in these areas here, but these areas called Jurujuba in Niterói. And in these areas, we've done releases through eggs. So we stored these buckets in people's house, and the bucket we put food for the larvae, we put eggs, and also we covered it. And after 10-15 days, the adults would come out of these holes here in the bucket. These are where we did all the release points in those areas that I've just shown to you. And here is the establishment of Mobakia. So we can have here year 1, year 2, year 3, and so since 2017. And what we see here in this green curve here, line, is the samples that are positive for Mobakia. So you see here that's being maintained. Remember, we stopped releases in these areas in January 2016. And since then, it's been stable. Mobakia is still there in those mosquitoes. And for me, is the most important point of this program is that to be self-sustainable. So 5 years, we don't do anything, mosquitoes are still there. In Australia, there are areas with 10 years after releases, and mosquitoes are still present in those mosquitoes. Wolbachia is still present there. We want to know if, like, after some time, after we stopped releases, Wolbachia density, like the presence of Wolbachia in those mosquitoes in the field, collected in the field, would have more or less Wolbachia. That could have a reason, would impact on the blocking on the virus. But what we saw was the opposite. So here, this graph shows the Wolbachia density here, the relative quantitation here. We've done this in 2 time points, 4 months after we stopped releases, and 14, 16 months we stopped releases. The gray here are mosquitoes that were collected from the lab, from the insectary where we do the production, and in green are mosquitoes at the same time collected in the field. So after all the process of integration and invasion, we collect these mosquitoes from the field. So you see here that Wolbachia present in the mosquito was higher compared to the lab. So that was true for 2 to 4 months, and also later on to 14, 15, 16 months, we see also significant difference, more Wolbachia being present in these mosquitoes. When we tried to see collecting these mosquitoes in the wild, bringing to the lab, and then challenging them with Zika virus and dengue virus, what we saw. So these graphs show you the RNA copies of the virus, like Zika or dengue, in the mosquitoes' tissues, like the head and the thoraces of these mosquitoes that were challenged. And we did with 2 different mosquito populations. One was collected from Urca, is a neighborhood in Rio that had no Wolbachia being released there. And the other is Jurujuba, the ones that I just showed to you. And what we see here is mosquitoes that have Wolbachia, they don't have almost nothing Zika virus in these mosquitoes. We did 2 experiments, they show more or less the same thing. When we did with dengue virus, the same results. Mosquitoes from Urca, no Wolbachia here, most of them are infected, but when we tested mosquitoes from the locality we've done releases, they are here much less infected compared to the ones with the other area. So these experiments were done 1.5 years after we stopped all the releases, and so mosquitoes collected from the field. Since at the end of 2016, the Ministry of Health in Brazil funded us to have to go much bigger. Remember, I showed to you the pilot studies in Tubiacanga and Jurujuba, and then we would expand to Rio and Niterói, and the total population covered here is around 1,300,000 people. So that's all these colored areas here. And very challenging for us is that 20% of all these areas are like this. So this is the Complexo Alemão in the north part of Rio, and we have also Maré. In Niterói, we also have communities like that, very complicated, very unsafe to work. So we had to really train our team members. Sometimes even the health agents were not able to get in these places. So I'm going to show you some results. They are a little bit not new. They are results from March last year, so 1 year ago. Why is that? Because of the pandemic, we have to stop monitoring to get into people's houses. So these are results from 1 year ago. So this colored the map here shows you what? Where in green shows area where Mobakia is above 60%. When we see that happening, If it's 6%, what we see, that's that Mobakia grows, and then established very well, and we have to do nothing anymore. In areas that are in yellow, means Mobakia around 40 to 59%, and in orange, less than 39%. So we are observing this. We have some new data on monitoring here through ovi traps, and is now being analyzed. For Rio de Janeiro, in the islands, we have areas that Mobakia is established in green, in medium, in yellow here, and other areas like those that we have all those important communities, those favelas there, Complexo Alemão here, in Bom Sucesso, Manguinhos, Maré. We see that Mobakia is taking more time to establish compared to other areas. So we are still doing some releases there just to be sure that we are going to have Mobakia in, and so they can protect the population. In areas that if we keep Mobakia low, like 20-30%, and we do nothing, what happens is that Mobakia will just collapse and will not be established. It's very interesting. There are lots of modeling being done around this. Epidemiology. So we want to know and also follow what is going on with arbovirus cases, which is transmission of dengue, Zika, chikungunya in areas that we are working. So this is a preprint of one of the results we are under review at the moment. What I'm going to show you some results from Niterói. Niterói was divided in 4 areas. So these are all implementation areas, the colored areas, 1, 2, 3, and 4. So that was the sequence of the one that we implemented, first year, second, third, and fourth. And this is our control area. Was not chosen as a control initially. It's just an area that we've done anything so far. So it's no Mobakia is there at the moment. Just for you to know, in zone 1 we had only 23,000 people, zone 2 68,000, zone 3 178,000, zone 4 101,000 people, and the control zone 111,000. And these are in the area in square kilometers. And these are the estimated number of released mosquitoes in these areas. Here you can see the maximum we released in this area here was less than 13 million mosquitoes over a period of time. So this is the Mobakia establishment, mosquitoes with Mobakia in zone 1, zone 2, zone 3, zone 4. So this was the first one, so Mobakia going up and being established. Zone 2, medium. Zone 3, a little bit less. And zone 4 started much later on, and we had this decline here. Is an area that we are observing. Maybe we need to go there and release more mosquitoes. But so that's the Mobakia establishment. So this is the follow-up on dengue cases happening there. Maybe it's a little small for you guys, but what you can see in each graph is the incidence of dengue, which is the number of cases of dengue per 100,000 people. So it's called incidence. So that was the historical data since 2007 up to 2020. Here you can see zone 1, 2, 3, and 4. And then these shaded areas here is the Mobakia establishment in each one. It's more or less what I shown to you in the other previous slide. What I want you to see is the right graphs here. So the scale is a bit smaller. You can see better the results. What you can see, so the dotted line shows the dengue incidence in the control area. in the full line here in blue line shows the incidence of dengue in that particular zone. Zone 1, zone 2, so this is the control, zone 3, and then zone 4 here. They are very similar, and because Mobakia was established later. What you can see here, that Mobakia being established, you have less dengue happening. And the numbers show a reduction of 44 to 77% less dengue when Mobakia was established, which is very good, very good results. We are very optimistic around these results. If you talk about chikungunya, there was an epidemic of chikungunya in Niterói. Again, zone 1, zone 2, zone 3, zone 4. So this is the chikungunya incidence, number of cases per 100,000 people. And then chikungunya historical data is much shorter, since 2015. Remember when it was introduced in Brazil. And then this is incidence in the control area compared to the in Niterói. We see here much less in the zone 1, zone 2, zone 3, and this is the control area, which is similar. No, sorry, this is the zone 4, but the difference is here. Mobakia is in the shadow area, the shaded area. Reduction is around 60% for chikungunya, which is still very important. So we are very also optimistic for chikungunya as well. And we are also very optimistic after these results. This is a result coming from Indonesia, another country that participates in the program. There, they had a randomized controlled trial. For epidemiology, is a golden standard study that was done in that country. So they did a study area of 312,000 people. This is the area, and they did. This area was randomizedly chosen to receive Mobakia in those blue areas here, or clusters that did not receive Mobakia, and they checked in primary care clinics the hospitalization of dengue patients. So that was done by dengue. What they found that areas that received mosquitoes with Mobakia, they had 77 reduction in dengue incidence compared to areas that did not receive the program. Again, very similar to what we got and very promising results. Right now, we are expanding this to other cities in Brazil. So I saw showing the map. I talked about Rio de Janeiro here in Niterói here, covering 1.3 million inhabitants. We also started already Campo Grande with around almost 900,000 people, and Petrolina will start more mid of this year with 343,000 people. In Belo Horizonte, we don't know the population to be covered yet because we are doing a study similar to what was done in Indonesia, and I'm going to explain to you. So this is the study in Belo Horizonte. Belo Horizonte here this is the map. Was 58 clusters. These shaded areas here in gray. They were designed around public schools for kids of around 6 to 11 years old. And so the 58 clusters, 29 will receive mosquitoes with Mobakia, so it's an intervention, and 29 will serve as a control. And this will be followed for 4 years. In these areas, 3,500 children were recruited for the study. The study will be conducted by the University of Minas Gerais, is another group. Our role in this is just to have the intervention done to release mosquitoes with Mobakia, be sure that they will be there, and then they will follow up this study for 4 years. What's the objective to see? What we expect is to see areas that receive mosquitoes with Mobakia will have less incidence. The children will have less response to arbovirus, dengue, Zika, chikungunya, compared to the areas that kids are not protected, the control areas. So this is just examples of engagement activities we've done in Belo Horizonte. Everything was done mostly last year with the pandemic, everything virtual, lots of Zoom calls, what we've done, you guys also studying, doing classes, taking classes in lectures through digital platforms. We engaged 114,000 people was done by both our team and also municipality team. So 430 activities with health agents, local leaderships, and also schools, directors, and so on. Every time before we do the releases, there is a pre-release survey. So we have people that go in the street. So this is an example of a health agent. She's doing a survey here just to ask the people if they approve the release of mosquitoes. So we want to know if the community is okay with that. So 95% of the Mobakia, of the people that were surveyed of these 348 understood the method, and 87% agreed with the mosquito releases. So if we put this together, understood and agreed, is around 90%, 90, which is very good, very similar to what we got in Niterói and also in Rio de Janeiro. So this is just some photos of the biofactory in Belo Horizonte. This is the first example of one city that built by themselves the facility. So they refurbished an old health clinic from the city, and so we are working there producing mosquitoes to be released in Belo Horizonte. So, which is great, a very good partnership done with Belo Horizonte. And I want to emphasize here, I'm almost done, almost finishing here. Very importantly, we all our engagement with the community, also with municipal agents, also stakeholders, everyone, we talk about that Mobakia, the Mobakia method is complementary, is not the solution of everything. It has to be used in conjunction, integrated method. You guys know entomology in agriculture, integrated pest management is very important here too, to be done with other strategies to make sure that we are gonna be successful. What we want to have is less mosquitoes, people taking care of their yards, reducing mosquito breeding sites, but Mosquito is our mosquito with Mobakia that we call, is there to protect people. I want to thank the funders and partners, the Fiocruz and also the Ministry of Health in Brazil. Since the beginning, we got funding from them since 2012. The Bill Gates Foundation as well as a great partner since the beginning. And we had also some support of funding from CDC for the epidemiological studies in Brazil. And our great partners and community members, the municipality health and education sectors. Thank you very much for listening to me. And if you have any questions, please visit our digital platforms, Facebook, Instagram, and also YouTube, where you can find updates, information, and lots of contents, WMP Brazil. Thank you so much, and I'm here to answer your questions. Thank you.
Thank you so much, Dr. Luciano. At this point, we will move on to to questions asked by the public. Let's see the first one. It's from Carlos Neves. Good afternoon, Dr. Luciano. Are there plans to apply these Mobakia mosquitoes in other Brazilian regions, such as the North and the Northeast?
Thank you for the question, Carlos. Yes, the Minister of Health is really wanted us to go to other cities. We are already going now in the middle of the year for Petrolina, which is the northeast part of Brazil, but the idea may be to go to Fortaleza, to Manaus, and also maybe in the south of Brazil, maybe Foz do Iguaçu. So this is something already in mind, but negotiations are ongoing to have funding available and everything. So we want to, this partnership with the Ministry of Health is long term and it's very important.
Thank you, Dr. Luciano. The next question is from Jessica Martins. She asks, have you ever had to deal with some kind of resistance from the population that due to the lack of information, they think that releasing a bacteria into the environment can be totally harmful?
Very important question, Jessica. Indeed, I think that is really solved with a good engagement, with lots of information. We know Mobakia is naturally found, as I mentioned to you in the beginning, at least in 60% of all insect species. So it's really there everywhere. We eat Mobakia because it's present in the bugs that attack maybe beans, rice, and also flowers. So it's very common. And also common that in mosquitoes like Aedes aegypti and Mosquito, and Culex mosquito, so they have the same inbreeding sites, and people are bitten by Culex for so many years since you were born. Culex, the normal mosquito that comes at night, they have Mobakia. So it's a question of explaining, and we know if mosquito dies, Mobakia will die as well. It doesn't live outside the cells, which is good at that gives more protection in terms of somebody has a would think that would be harmful to the environment. So that's what we do. Inform, show lots of data in terms of studies that is natural and safe.
Thank you, Dr. Luciano, for the answer. The next is from Frederico Salles. Do you have any idea of the dispersion capability of these mosquitoes? When you release mosquitoes in one area, what is the chance of these mosquitoes to disperse for other areas?
Very nice question, Frederic. Thank you. Indeed, when we started with those pilot studies, we installed BT traps outside, and the ones that were maybe 1 kilometer from where we did releases, we never found any mosquito with Mobakia. So Aedes aegypti is known to be really, they like to be close to humans, so they don't fly much. They fly around 250 meters, and if there is wind, maybe they go. But what happens if mosquitoes with Mobakia, if someone takes from one place and takes to Viçosa, for instance, they will be there, but Mobakia will not establish because they need this time to really to multiply, to cross with the mosquitoes there, and really to be able to progress to that particular population.
Our next question is from Lucas Promantachoki. Is the introduced mosquito more competitive than native mosquito species?
Lucas, we do as quality control some lots of experiments in the lab, and also we did some in the field. Actually, they are very similar. The difference is that they have Mobakia. What happens when Mobakia is present depends on the strain of Mobakia. The eggs cannot be dried for long periods of time. Maybe there is some fitness cost which is minimum, like around that will around 10% that will have this impact, but they are not more competitive. What makes them more competitive, I can say, is the Mobakia there, because Mobakia, because they manipulate the reproduction. I didn't have time to explain, but there is a phenomenon called incompatible cytoplasmic incompatibility. When a male with Mobakia cross with a female that does not have Mobakia, that female becomes sterile. So females that have Mobakia, they have more advantage. They produce eggs with Mobakia. All the siblings will have Mobakia, and then Mobakia makes sure that goes to the next generation. So that's the fitness advantage, I would say.
Thanks again, doctor. The next from Frederico Salles. Why don't you release larva or pupille with Wolbachia?
Very, very good, Federico. Actually, this was trialed in Vietnam by doing pupae, but because we are, as I showed to you, we are doing at the moment a huge expansion. It's very hard. So the best now we are doing will be to do egg releases, which you can just dry and ship to other cities. So you have a facility that produces millions of eggs, and then you can just ship these eggs. But that was tried with pupae in Vietnam, but in terms of logistics was not so successful. The best is, I would say, eggs, adults, and then the other pupae and larvae.
Thank you, Dr. Luciano. Next question from Edwin Dominguez. Do you need permits to release Wolbachia mosquitoes in a community?
Yes, we do. We had first we had a permit from IBAMA, which is the Environment Ministry in Brazil, to import mosquitoes with Wolbachia, the eggs from Australia, because they were originally Mobakia was originally introduced there in Australia. And we also have permit from Anvisa to be able to release mosquitoes with Mobakia in the wild. So because we release males and females. And also we have permit from CONEP, which is the Human Ethics Committee, the National Human Ethics Committee, because we release mosquitoes that bite people. So we have all these permits. And also, of course, the community, they permit us to release. Of course, we have a consensus, we have this engagement, and they are okay with the release. We never had any resistance in terms of community members say no, or movement that people that would say don't release.
The next is from Lucas Arcanjo. What factors you guys consider to determine the number of mosquitoes to be released in some area?
Very good question, Lucas. So first we had more or less, we have a grid, and then we around 50 mosquitoes per grid, which is around 150 mosquitoes every 50 meters. So that was but now we are kind of calibrating based on the population of specific areas. So we want to do more in terms of population. Of course, there is a correlation in terms of human population and mosquitoes. So if we have more wild mosquitoes, we have to release more mosquitoes with Mobakia. So that's a calibration we are doing at the moment.
Next question is from Professor Simon Elliott. Considering your response about dispersal, it will need to be introduced everywhere in every municipality. This seems like a big problem, yes?
Thank you, Simon. Good for you to be here. Yes, so that's one thing that we will have to do if we want to do to many places is, of course, we cannot do everywhere at the same time. We have to have phases that we can grow and going to other cities. We are already discussing how we will operate in the future. Of course, we will need to have partnership with NGOs, with of course continue with the health agents in each city to be able to really to expand and introduce the program in different cities. Partnership, that's the word.
Thanks again for the answer. The next is from Douglas da Silva Ferreira. About dispersion, why in this methodology do you release males and females? Why was it possible to just release the males?
Very good question, Douglas. Yeah, actually, there are two methods using Mobakia. There are some groups working with males. So what happens if we do only males? You have suppression. So remember, I explained to you the cytoplasmic incompatibility. When a male with Mobakia crosses with a female that does not have Mobakia, that female becomes sterile. What they do, this group, so the suppression group, they release males, and then the idea is to crash that population. But of course, this will work as very similar to insecticides. You do for a while, and then you reduce population, but then you have all the egg bank there that eventually will come back, and then you have the population back again. In our case, we want to have integration. We want to have this sustainability. So that's why we release both males and females, because Mobakia, the females are the ones that make Mobakia to disseminate to over many generations. I'm not sure if you understood my answer, but some people also call our method is the replacement. We substitute the population that would transmit most diseases like dengue, Zika, chikungunya by a population that will protect that population, that human population.
I think we don't have any more questions.
Yeah, we have some comments. Simon says, congratulations on your work, Luciano.
Thank you.
Gabriela de Figueiredo says, the potential of this work is incredible. I hope that this methodology will spread to other regions. Thanks for the talk, Dr. Luciano.
Thank you, Gabriela.
I think that's all.
Okay, so we thank Dr. Luciano for his brilliant participation. People are thanking for your talk in the YouTube chat. And now we are ending this afternoon broadcast. We inform that this video will be available on our YouTube channel. And we have important information for you that's from the entomology program at the Federal University of Viçosa.
Colleagues, we're selecting new members for our group of studies. We'd like to invite you to join us. Feel welcome to participate in the selection process of Insectum. We thank you all for attending this event.
Thank you all. Thank you for the invitation. It was an honor to be here with you guys. Thank you, Insectum.
Thank you. Yeah, good afternoon for all.
Bye-bye.